Okay, great. So maybe we can start so we can hear what Dr. Hantman has to say about us. So I have to say it's really a pleasure that Adam accepted to talk to us. For years, I'm trying to convince my colleagues to invite him to talk to us. Unfortunately, he couldn't visit us, but you know, I did extend an invitation for him to come and see us whenever he wants, and it's really anytime, Adam, you want, you are welcome here. So in terms of introduction, you know, because of the web, you can find anything you want these days. I highly recommend that you do a Google search, Adam Hantman, Janelia, and will take you to a page where describes you know, the people when he was at Janelia Farm, and practically gives you a backstory of his career. I'm not going to read it all. I'm just gonna start from where he came to my radar screen. So I remember his paper when he was in Jessel's lab, Tom Jessel's lab back, I think, 2010. And he had a really nice paper on the column of Clark and proprioception, you know, and I thought was really well done. You know, the mouse he described, the GDNF, a reporter mouse for Douglas of Clark, you know, still is used. So since then, I have followed his work really closely. Before that, before he came to Tom Jessel's lab, he worked with a great neuroscientist, in, you know, one of the founders of the Society of Neuroscience in 1969, Edward Pearl. Edward Pearl has worked for a long time on nociception, and is where he, Adam, you know, started, you can say, real scientific career, quotation marks. Then after he went to Janelia Farms, he continued his work. He published, I think he published the, for me, I don't know, people might disagree, the most interesting work when it comes to motor control and proprioception in general, the last five years. So I highly recommend that you go and look at the PubMed and his papers. I think I talked enough, and I think we should let Dr. Hantman say what his thinks. I think this will be an exciting seminar and really instructive to us, something we can learn and maybe take some lessons what to do next. Thank you.
Thanks so much, Pantelis. I really want to extend my gratitude for asking me to give this talk. I'm really excited. I still have a deep love for the spinal cord, and I think the mission that you guys have is super important. And so I'm really excited to talk with you all today. During my morning sessions, some of you shared your screens and showed me that it was 80 degrees in Miami today. So it's a little upsetting I'm not there, but it's still quite nice here in North Carolina. So I don't feel that bitter about it. And as my introduction might have suggested, my lab is interested in motor control. Actually, I think the overarching interest that at least I have is in complex systems, and really how sort of interactions of all the parts of a system are give you functions that are greater than the sum of all those parts. And I think that's what brings us to the nervous system. I think it's one of the most beautiful complex systems that we know of. So that brings us to the question of why motor control. Well, I think motor control is one of the essential functions of the nervous system. It's really probably why we have a nervous system, at least evolutionarily. It's really involved in all aspects of our survival. It's how we find our food. It's how we find our mates. It's how we avoid our predators. Now, it's true that you don't actually need a nervous system to produce movements. Obviously, single cell organisms move through the world, and even very simple animals like this trichoplax here, which has no discernible nervous system, still goes through the world producing behaviors in order to survive. So what I think the nervous system allows us to do is to upgrade to much more complex behaviors. It allows us to sense and respond to things from much further away, I would say like vision. It allows us to move faster with more control, more coordination, and it allows us to use really large effectors such as our limbs and even tools. So the question then becomes, how does the nervous system afford us these upgrades? And let's take limb control as an example. This is something sort of near and dear to the heart of my lab. And what the nervous system must do to control something as complicated as a limb is understand the properties of that limb. And then given the understanding of those properties and with some goal in mind, ultimately what the nervous system has to do is pretty simple. It just needs to generate a pattern of activity coming out of its motor neurons, which will influence the muscles in a particular way in order to hopefully move the limb in a fashion that accomplishes the behavioral goal. And so quite simply, you could think about everything upstream of the motor neurons, as in the rest of the nervous system, is their main role is to generate patterns that will generate the patterns in the motor neurons. You can think about this as a pattern generation problem. And actually, many people through the history of neuroscience have thought of this problem as a pattern generator, really going all the way back to Charles Sherrington, who was really interested in the pattern generation systems underlying rhythmic movements such as locomotion and scratching. And actually, quite interestingly, Sherrington thought that for locomotion, for example, the pattern generations were set actually not by the nervous system, but actually by the periphery. That actually the sensory information coming back into the nervous system had the information about the next step, and all the nervous system had to do was convert that sensory information into the motor output. Now, this was convincingly demonstrated to be incorrect by Sherrington's own student, Thomas Graham Brown, who showed that actually pattern generation was central in origin. And actually, Graham Brown actually showed that the spinal cord could generate the basic pattern of activity for locomotion all on its own. You could deprive it of its descending information, deprive it of its sensory information, and as long as you gave it a kick, it could generate the basic pattern of activity required for locomotion. Now, my lab is also interested in this sort of ability to generate useful patterns, but not for these rhythmic movements, but for skilled movements. These are our learned movements. These are the ones that we use to interact and manipulate with the world. It's the ones we use to feel the world around us through touch. It's the ones that we watch, pay a lot of money to watch other people do. And the reason why I think we're so interested in it is these pattern generators have to be learned. They have to be acquired. And we're interested in how that happens. And because of the role of skilled movements in our lives, many neuroscientists throughout the last 100 years have been interested in the neural basis for skilled movements. And they've been sort of marching around the entire central nervous system and trying to assign function to all of these different regions for skilled movements. For example, cerebellum, which is an area that my lab works on, has been shown to be involved by us and many other people in the fine-tuning of these behaviors, allowing us to be masterful at these behaviors. And we have a nice story that came out a couple months ago that I won't have time to tell you about that demonstrates maybe why cerebellum is involved in that. And I won't tell you about the cerebellum today because today we're going to focus on another region that has garnered a lot of interest for skilled movements, and that is cortex, and more specifically motor cortex. And this is a region that, really we think is sort of a master commander, at least in primates, for performing skilled movements. This is a region that when you damage or disease it, it leads to real, almost selective loss, at least in some species, in our ability to do skilled movements. This is the region that when you stimulate specific parts of it, you'll actually get very specific movements of the musculature. And this is also the region whose single neurons in the population at large will exhibit patterns of activity that are highly related to features of skilled movements. And I think one of the most sort of instructive views of that comes from the work of Krishna Shenoy's lab, who's been looking at motor cortex population activity and how it relates to skilled movements. You don't need to understand these plots just yet, but just recognize that every one of these trajectories, every one of these lines moving through the state space plot represents what the motor cortex is doing for many different classes of movements. And all I really want you to understand here is what it looks like is that motor cortex has a separate pattern of activity for every one of these different behaviors. And so it might seem actually like we understand quite a lot about how motor cortex might be involved in something like a skilled movement. But I would actually argue that there's just many really basic questions that remain really wide open. And we're going to try to take on one of these basic questions today. And that basic question is, where does this pattern of activity that we're seeing here come from? Okay, so there are two possibilities of where this sort of pattern of activity, our underlying skilled movements, comes from. The first is it comes from motor cortex itself. That motor cortex over the training period has changed its synaptic weights in a way that it can generate the basic pattern of activity that's required for the movement on its own. All it needs is a kick from some other part of the nervous system in order to initiate this thing, but then it can handle the generating the pattern. This is like an autonomous dynamical system, basically. And actually, I would argue that this is sort of the lead hypothesis in the field. But there is another alternative hypothesis, and I would actually argue an older hypothesis, which is that motor cortex is critical for generating these patterns of activity, but it actually can't do it on its own. It actually requires information from many other regions to come together, get integrated, and then from that integration, the correct pattern of activity will emerge. And this is sort of a distributed pattern generation system, or sometimes known as an input-driven pattern generator. And I think actually discerning between these two different hypotheses is really, really important. There's a lot at stake, not only at stake at the nature of the pattern generator itself, but it also tells us how we should move forward with experiments and theory. For example, if you're sitting in this autonomous mode and you really want to understand how you generate patterns for skilled movements, but you should study the fine features of motor cortex. But if you're in this more distributed, input-driven system, then really the work is trying to understand how motor cortex interacts with all of its inputs in order to construct the dynamics that are so critical for movement. All right, so how do we get here? How are we, 100 years later, and we don't know this very basic answer? And I would argue that one of the reasons is that most of the pioneering work in this field has been done in primates, both humans and in non-human primates. And that's been, absolutely revelatory. These species are excellent for doing recordings, for doing excellent behavior. But where they're a bit more limited is our ability to do causal perturbations, where we affect some part of the nervous system, and then we see what the consequences both neurally and behaviorally. And I think it's those classes of experiments that have real promise of disambiguating between hypotheses, like the one that I have laid out as an example. And so one of the goals of my lab, really beginning when I moved to Genelia, whatever that was, now 12 years ago, was to actually port these questions down to another model species where we could actually do those classes of causal perturbations. And the model species that we selected was the mouse. We selected it for a couple different reasons. First of all, the mouse makes excellent use of all these new fantastic tools for doing causal perturbations, tools like optogenetics, tools like pharmacogenetics. They were in part developed in the mouse, therefore they work quite well. But we also picked the mouse because mice can perform very nice skilled movements, and perhaps the champion of skilled movements in the mouse, at least for the neuroscience community, is this task. It's a reach to grab to eat task, really pioneered by the work from Ian Wishaw. And the way that this is typically done is you take a freely moving rodent, you put it in a little box, you have the slit in one end of the box, and then when the animal wants the food pellet, he runs up to the slit, reaches his hand out, grabs the food pellet, and places it in their mouth. And one of the sort of thing that always gets me about this behavior is that if you compare how a rodent does this to how a human does it, they're very, very similar. There's sequential steps that look very analogous across these two species. So we really liked this behavior because of this analogy to our own behavior. But there was one thing that we wanted to change about it, which is that we wanted to convert this traditional freely moving task into a head fixed version of the task. And there's a lot of advantage for head fixation. First of all, it allows you to isolate the movement as much as possible to the effector that you're interested in. It allows you to impose cued trial structures onto the animals, which is a huge benefit for the work in the primate. And most fundamentally, and I think the reason why we really did it was it really facilitates these recordings and manipulations that we had planned to try to understand the neural basis for skilled movements. And so over the last 10 years, we've been developing this new behavioral assay. A head-fixed animal sits in this little comfortable rig. There's a table out in front of the animal. And after a tone and a prescribed delay, this table will rotate, deliver a pellet of food within the animal's reach, and then hopefully the animal will reach out, grab the food pellet, and place it in their mouth. Now, there's one thing I need to tell you about this behavior, which is kind of critical, which is that it's basically done in the dark for the animal, or at least very, very low light. And so the way that we think the animals are solving this task, and I don't have time to go into the details about this, is that they are memorizing this position of the food pellet and then storing that memory within their nervous system and then calling it up whenever we cue them. So after training, which can last anywhere from a couple days to a couple of weeks, depending on the trainer and the trainee, these animals really get this behavior. They understand the tones and the prescribed delays. They can smoothly and coordinately execute through all the steps of the behavior, and they can get quite good at this behavior. So if you even look at the first attempt of a trial, these animals can achieve success rates on over 90% of the trials. These are just a series of trials. I didn't cherry pick these. These are just one after another. These animals can just crush this task. They're the sort of Michael Jordans of the reaching task. Okay, so we really like the level of skill that these animals can achieve. But there is one problem with this behavior, which is that basically it's represented by raw video. There's no sort of analog readout of this. And actually, we realized that quite early on, if this was going to be the bread and butter of the lab, this was going to be a real bottleneck for experiments and for analysis. Now, this was around 2012 that we were starting to develop this task. And at that same moment in time, the computer vision machine learning world was exploding with these techniques to analyze at least images using machine learning approaches and computer vision approaches. And we thought that perhaps we were in a really good position to do that because it's a very simple behavior repeated over and over again, and we have good camera with high resolution, both in space and time, and sort of nothing ever changes. So we thought that we could solve this with the computer vision approaches. So we teamed up with a brilliant computer scientist named Kristen Branson, who's at Genelia. And over the last 10 years, we've been developing a suite of tools to analyze all the different features of these videos that we were interested in. And so one of them basically will convert the videos into ethograms, or graphical depictions of the movement, telling us when all these different steps of the movement occur over time. And basically you can make a little ethogram out of those. That's very useful. And we also have other approaches where we can track unmarked parts of the body in 3 dimensions where the animals are performing this behavior. And these are also now been replicated with techniques like deep lab cut. Okay, so this is really fundamentally great. It changes how we do our analyses, but it also changes how we do our experiments, because unlike humans, these classifiers and the tracking assays here can actually work in near real time if you engineer them correctly. And that allows us to close the loop with the animal's behavior, where we can detect features of the behavior and then trigger back experimental perturbations in real time. And you'll see in a few slides that ends up to be critically important. Okay, so we were feeling pretty good about our behavior here, and we wanted to now go figure out how motor cortex was involved in the skilled movement, but we were sort of confronted with a sort of major problem, which was around the same time, the field was also losing faith that rodent motor cortex was really doing the same thing for skilled movements as was a primate motor cortex. And a lot of the reason for this doubt actually came from chronic lesion experiments. So if you remove a very large chunk of cortex, even beyond motor cortex, as is done in this rat, the animals can still do the behavior. They can do all the steps. They can actually do it pretty well, but they do fumble the food pellet at the end, and they do have some problems with supination. And I think that these chronic lesion experiments are really, really interesting. And I think one thing that they can teach us about is what the rest of the nervous system can do to compensate for the loss of this region. And I think that's very, very interesting. And actually, my lab has a big fraction on working on sort of similar issues. But really what we wanted to know is, what is the normal region of motor cortex in an uncompensated brain? And I think if one wants to answer that question, one way to do it is to try to take, yeah, is there a question? Sorry, I thought I heard somebody. One way to try to get at this is to try to find a way to take motor cortex off while minimizing compensation. And of course, we're in the era of optogenetics, and optogenetics is really ideally suited for these kinds of experiments. We can do random, unexpected, acute perturbations and then reverse them. Okay, so that's what we wanted to do. We wanted to try to take motor cortex offline using optogenetics really for the first time for these reach tests. So the way that we're going to do this is we're going to really capitalize on a mouse line that was made by Guopin Feng at MIT. It's a mouse line that expresses channel rhodopsin in the inhibitory neurons of the cortex. Expression of that opsin and exposure to blue light activates those inhibitory neurons, and then that co-ops their normal function, which is to sort of shut down neurons locally in cortical areas that they are sitting in. And so what we're going to do is we're going to sort of, in the beginning, sort of take a sledgehammer here. We're going to turn the blue light on throughout an entire trial, but we're going to do this on random intermittent trials so the animals can't predict it. And then we're going to try to explore a variety of different cortical areas and see what parts of cortex were involved in this skilled movement. And we tried many regions, and most regions actually gave us absolutely no effect. And I'll show you one example of that. This is ipsilateral sensory motor cortex. The laser comes on at the beginning of the trial. There's a cue, the pellet's delivered, animals do the task perfectly normally, no issues, okay? Now, this is ipsilateral sensory motor cortex, so you might not have imagined there'd be much of an effect. But I think it serves as an important control that if you ever do see an effect, it's not from the blue light or some non-specific effect of shutting off a large swath of the brain. All right, so IPSI doesn't have an effect, but what about contralateral? We expect many regions might have an effect. Again, most regions gave us absolutely no effect on the behavior, even the fine features of the behavior, but that was with one major exception, which is contralateral sensory motor cortex. And here the effect was dramatic in that if you turn this region off throughout the duration of an entire trial, the animals would simply never initiate the behavior. So this is either a really boring movie or a really exciting movie. The laser's on, motor cortex is off, we deliver the food pellet. And for basically as long as we leave this laser on, the animal's simply not going to make any movements. They're never going to initiate the behavior while motor cortex is off. So we were a little bit worried that this might not have been a consequence of the arm movement itself, but just that the animal wasn't understanding the trial structure. So we decided to vary this behavior by keeping the trial structure exactly the same, but allowing them to solve the task with a simple ballistic movement of their tongue, just to lick the food pellet. So we raise the food pellet up, turn motor cortex off, and see if they still understand the trial structure, and they do. They're able to complete the entire task. All right, so this really suggested that we needed motor cortex for the initiation specifically of this skilled movement. But I want you to notice something about this movie, which I think is just really important to really watch every movie and understand the fine details. Because watch what happens here. The laser's on, he's going to actually lick for the food pellet, but watch what's going to happen here. This is the normally affected hand, and he's actually going to initiate a movement of his hand, even though the motor cortex is off. Now, this movement is not that different than the normal learned movement. They're going to lift, they're going to supinate, they're going to go to their mouth. They're just not going out to this remembered position in the world. And here, motor cortex is not necessary for the initiation of that movement, further speaking to the real requirement of motor cortex specifically for this sort of learned skilled movement. All right.
Adam, can I ask a question?
Sure.
Why do you think they do that? They got the damn thing in their mouth already. What do they need to make those movements for?
To help the food pellet, you mean? Why are they moving the? So sometimes, especially sometimes they'll actually remove the food pellet, chew, put it back in. So they're actually doing a lot of manipulation of the food pellet in their mouth. And it's very interesting. Like in other work, we have what Ian Wishaw also started a pasta task where they like chew a piece of pasta. And there you can see them really like, helping get the food pellet onto their teeth. So yeah, I think it's more than just the food manipulation, which is a very sort of ethologically sort of important and kind of innate behavior.
Thanks.
Yeah. Okay. So motor cortex seems to be required specifically for this initiation of this learned skilled movement. But is that all you need motor cortex for, or do you actually need it for the continuous execution of this movement? This has actually been a really long-standing question in the field of motor cortex. and motor control more generally. And it's been long standing because we haven't been able to do one of the essential experiments, which is to let the animal start a movement totally normal without any perturbations, then detect that start of the movement, and then trigger a perturbation midway through the performance of an action. And that hasn't really been possible before, but with our closed loop nature of our experiments and the rapid onset of optogenetics, we're able to do that experiment for the first time ever. And it was sort of an amazing moment in lab, because it sort of gave us like superhero level status over these animals, and that we could stop or pause them almost anywhere along their length of their reach. So let me just show you a couple examples of that. The first example, we're going to actually try to stop motor cortex during this first phase of the movement where they're going from the lift to the grab. We think of this as a roughly ballistic movement free from a lot of sensory guidance, thought to be pre-planned, at least in large part. And what we notice is that if we turn off motor cortex midway through that behavior, what you'll see is the hand will immediately stop and either hold in midair or retract back. So it is apparent that motor cortex was needed for at least this ballistic pre-planned version of the movement. But what about after you start to get sensory feedback? Do you still need motor cortex, or actually can the sort of sensory information take you all the way home through subcortical systems? And so in order to answer that question, we're going to hold the perturbation off all the way until after he grabs the food pellet and has the food pellet in his hand. He's about to do his supination steps. And what you'll see here is, again, he's not able to complete the task. So we're going to turn the laser off right there, and you're going to see he's no longer able to deliver this hand up to his mouth. And this is something we never see. An animal never gets paused in this moment here. But I want you to notice something else about this video. It's super important, which is that this animal is not disengaged from the behavior. He's actually trying to pull his affected hand up with his unaffected arm, as you'll see here as it replays. So hand stuck, and he's actually trying to pull this hand up with the other arm in order to complete the task. And what I want you to remember or think about here is that, yes, motor cortex is needed for the movement of this hand, but during that perturbation, the rest of this animal's brain still knows what's going on and is actually trying to figure out a new way in order to complete this task. And it's just kind of important to remember. All right, so again, is this specific for the learned skill movement, or is this true for any complex movement of the forelimbs? We thought maybe grooming would be a nice version of this task. Very complicated movements of the forelimb, but they're thought to be more innate. And what we find is that turning off motor cortex, which just happened there, has no consequence on the animal's grooming, not even a shudder of their response, even when it's midway through their performance. So this is starting to build up sort of a nice necessity story for motor cortex. It's necessary for the initiation, and it's necessary for basically all of the different steps, almost all the way up to consumption of the food pellet. So in addition to this necessity story, in these same experiments, though, we also started to get some hints about potential sufficiency arguments for motor cortex, and more specifically that motor cortex might be sufficient to evoke these behaviors. So let me just take you through what we saw. So what we had seen is that every time we had turned off motor cortex, once we turned it back on or allowed it to come back on, and almost every time in really well-trained animals, the animal would reach back out for the food pellet. So these are concatenated trials again. Laser goes from on to off, and every time when we turn the laser off, the animal reaches out for the food pellet. Now, maybe they remembered the food pellet, maybe they resensed the food pellet, but that didn't actually explain all of the cases. And there was something kind of funny about the timing of these responses. So if you look at the latency from when the laser went off to the production of this behavior, that latency was shorter and tighter in its jitter than was the latency between the cue and the normal behavior on control trials, which is obviously what the animal had learned. So that made us start to think that maybe turning off motor cortex and then releasing it was actually causing the behavior to happen. So we wanted to test this, and we thought that one way to test it was to take an animal, put them in the rack, give them no reason to reach for the food pellet, no cues, no food pellets delivered, just sitting there basically in the dark. And all we're going to do is turn off their motor cortex and then release it. And lo and behold, what we found is just that perturbation in its release caused the animal to perform the entire behavior. So cortex is off. we turn it back on, and then for no reason, there's no food pellet out there, they'll reach out for the food pellet. Okay, so this really is starting to suggest that perturbing motor cortex and then releasing it doesn't go back to resting state. It actually will go down the neural trajectory that will actually lead to the behavior. And so I think both this necessity story and this sufficiency story, at least for evoking the behavior, starting to suggest a real prime role for motor cortex in the rodent. Actually, you could argue that the evidence we have collected here suggests even a more important role than the evidence we even have in the primate. And that really justified to us going in and digging in on the mechanisms by which motor cortex are actually controlling skilled movements. Okay, so I set up this case earlier on in the talk that maybe they're involved in these different kinds of pattern generators. Well, I told you that motor cortex is important, but we actually haven't shown that there are actually patterns of activity in motor cortex of any kind during these movements in the rodent. And so we first decided to go out and make sure that was actually true. And in order to do that, what you need to do is record from motor cortex while the animals are producing the movement. And this is where we can really leverage the head fixed nature of our experiments to establish really nice high quality recordings. Basically what we do is we make a craniotomy above the motor cortex. Then we put in these high density silicon electrode arrays into motor cortex, and we're really targeting the deep layers of motor cortex, layer 5, layers that are going to go to places like the spinal cord, the brainstem, the other side of cortex. And then the goal is to try to record from as many units as possible at the same time. You can see a subsampling of the electrodes down here where I've colored in putative units in these different colors. So we're getting high quality, numerous simultaneous recordings from layer 5 of motor cortex. And you can see the animals perform the behavior. There's even some modulation of these neural activities around the behavior. Now that's a little bit hard to see in just this a few number of trials that are sort of passing you by here. So let's look at a lot more trials. These are 4 representative neurons, 1, 2, 3, 4 here. And what I've done is I put on histograms of their activity and then raster plots for every one of these little ticks is a spike from these neurons, and trials are on the different rows here. And what you can see here is real diversity of the response properties in motor cortex. So some neurons have this nice simple activation, increased firing rates around the behavior. These are all lined up to the production of the movement. Some neurons, interestingly, turn off around the movement, and then others have more complicated relationships. So there's a real zoo of response properties across these neurons, but within a neuron, they're pretty consistent. These are hundreds and hundreds of trials here, and you can see these neurons are largely doing the same thing trial over trial. All right, this is just 4 units though, so let's look at a lot more units. And what I'm going to show you basically is lots of these top plots represented as heat maps. So every row here is a different neuron. This is a heat map of their average activity pattern lined up to the production of the movement when the animal lifts. And what I've done is I've separated these neurons out into 3 major categories: those that increase their firing rate in relationship to the behavior statistically, those that decrease, and then another class that at least didn't pass the muster of our statistical test that we employed here. And what you can first see is the sheer fraction of neurons that seem to care about this behavior in motor cortex. It's basically 70 to 80% of the neurons. And remember, we're not hunting for neurons here. We just slide the electrode in and we take what we get. And we're not searching for anything. So 70% of those neurons care about the movement. A little bit over half increase their firing rate statistically, a little bit under half decrease. Okay, so the area fundamentally cares, but how do they care? Let's look at the neurons that increase their fire rates. What I've done is I've sorted the rows, which again are the neurons, by the timing of their peak firing rate in relationship to the behavior. And what you can see here is that some neurons actually have their peaks before the behavior even begins, but then others have their peaks that tile over the whole behavioral expanse. And so perhaps these are involved in initiation. And this is the reason why we can stop the movement at any time, because there's continuous commands coming out of motor cortex. And this is starting to give you the sort of impression that there's a real pattern of activity coming out of motor cortex in relationship to this behavior. Now, this is single neurons that we're looking at here, lots of them. But what we think is true for motor cortex, as in any region, is it's really operating as a population. And so what we really would like to do is look at what the populations are doing and see what their patterns look like. Now, that gets a little bit complicated, because population activity sits in very high dimensional space that basically is as high dimensional as the number of neurons you're thinking about, or in this case, recording from. And therefore, very hard to visualize when you're in 500 dimensional space. And so what most people do in order to think about the population activity is to do some sort of dimensionality reduction, and then they plot the dimensions that explain most of the neural variance. So principal components or other methods like that. And you can think about the lower principal components as like super neurons. They're like neurons, super neurons that reflect the activity of many neurons that you're recording from. Okay, so that's what we're going to do here. On the left, we're going to plot the 3 super neurons, the first 3 dimensions that explain most of the variance in motor cortex. We're going to plot them off for one another over all the different trials. So every one of these little lines represents a different trial. Okay, this is in the 3 dimensions of neural space, and then for relevance, I'm showing you what's happening to the arm in the 3 Euclidean dimensions of actual space for these same trials. And when I play this movie, what you're going to see is this beautiful activity coming out of motor cortex that over all of these trials really looks quite similar, suggesting that the population activity is really doing something very similar over all these trials. And we know that this pattern of activity is absolutely crucial. So if we deteriorate that pattern too far, the animal won't reach. If that pattern happens, the animal is going to reach. And there's rich information in these neural trajectories. Matter of fact, if you never showed me what was happening here on the right for the arm, I could predict where the hand was just from the neural activity. So we know there is rich patterns of activity in motor cortex for these skilled movements. So this brings us back to our original question of, okay, well, who's generating these patterns of activity? Is it motor cortex can generate it all on its own? Just needs an input to give it a kick? Or is motor cortex collaborating with all of its inputs in order to generate these patterns of activity? So, okay, the main difference between these two things is the role of the inputs. Here, the input is only needed as a kick, and here you continuously require these inputs in order to generate the correct pattern of activity. So the obvious thing to do is to manipulate the inputs and see when they're important and how they're important. Now, the major class of inputs coming to motor cortex comes from motor thalamus, just the regions of thalamus that project to motor cortex. And so we're going to take control of them. We've done this in many different ways now, but the original way we did it to suppress them was actually co-opting their natural inhibitory input, which comes from a little nucleus called the Thalamocortical nucleus. It's not little, okay, but this nucleus is called the Thalamocortical nucleus. This provides inhibitory inputs to all of thalamus, including motor thalamus. And so what we're going to do is put channeled opsin into the TRN, and then we're going to put a fiber that can shoot blue light out over motor thalamus. So the blue light will activate the inhibitory terminals, dump on GABA all over to motor thalamus, and that will suppress the transmission from motor thalamus to motor cortex. And the first thing we're going to do is just hit this thing with a sledgehammer again, take these things out for entire trials, but on random intermittent trials. And what we can clearly see is the animals cannot perform the behavior without motor thalamus. This is the probability of just lifting your hand off the perch in control, and when thalamus is inactivated, and you can see a real deterioration in the ability of the animal to even initiate the movement. So motor thalamus is clearly important here, but this actually doesn't disambiguate between our two hypotheses because thalamus could be important for initiation in either case. So the way to disambiguate is to hold back the thalamic perturbation until after the movement's been started, and then see if we can still actually turn off the behavior midway through the reach. So this is the trajectory of the hand in one of the directions, one of the dimensions of space. So this would happen on control trials. And then what I will do is I'll overlay what happens on these laser trials. That'll be a white line that turns to a green line when we turn on the laser. And you can see basically as soon as we turn off motor thalamus, the arm cannot continue along its progress. It either drops back down to its starting position, or actually in some cases will just hold in midair. All right, so motor thalamus is not just required for initiation. It is required for the continuous execution of this behavior, but this raises a kind of a sticky question of why. Because you could imagine a situation where actually motor thalamus is not critical for generating the exact pattern of activity. It doesn't have information about the pattern, but it provides a tonic permissive input to cortex. Without this input, cortex will drop below its threshold. And even though it has its own pattern generating capability, it will basically be floored and it won't be able to express its own pattern generating capability. So basically cortex will go to zero firing without its thalamic inputs. That's in contrast to the sort of integrative role of thalamus in generating the correct pattern of activity, where it has real information and commands about the pattern of activity. And without the thalamus, it's not that motor cortex goes silent, but it will generate the wrong pattern of activity. All right, so how do we test this? Well, what we need to do is record from motor cortex while doing these thalamic perturbations. And so that's what I'm going to show you here. We're going to go now back to population activity. This is now a trialed average population activity. Imagine all those plots I showed you before, but we're just going to average across all the trials because they're basically doing the same thing anyways. So this is what happens in control situations. When the animal wants to reach, cortex jumps out to the right in these state space plots. It bends around this angle, and after that, the animal will perform the behavior in the squiggly part of this line, okay? That's what happens normally. What happens when we turn off thalamus? Does cortex just go silent and just sits back here, or does it actually just generate the wrong pattern of activity? And what I think you can see here is that cortex, when we suppress motor thalamus, which is represented by green here, does not go silent at all. Matter of fact, kind of does the right thing initially. It jumps out here to the right in state space, and then it gets out to this part of the state space plot where things sort of fall apart for motor cortex, and it dives down into this neural abyss down here where it will perseverate for basically as long as the thalamic perturbation is on. And then if you release that perturbation, the cortical activity will jump back out to the right in the state space plot and then do the squiggly line, and you'll actually perform the behavior. So it does seem that cortex is necessary to generate the correct pattern of activity. So the flip version of that question, though, is, okay, if it's necessary, is it sufficient to steer around cortical activity? So for that, we did the contrary experiment, where instead of trying to silence thalamus, we're going to try to actually activate it. We're going to put channelopsin in motor thalamus, and we're going to shoot blue light onto motor cortex while recording from it. And we're going to do this at different frequencies, 4, 10, and 40 hertz, and see if we can steer around cortical activity. That's what I'm showing you down here on the left. This is neural activity. This is the hand position. And these are the different frequencies of stimulation, where the white line represents what happens on control. And what you can see here clearly is that these different frequencies of thalamic stimulation to motor cortex steer around the cortical activity in almost a frequency dependent way, where 4 hertz gives you a little blip, and then the 10 and the 40 hertz puts you in a very weird part of the neural space. And therefore, the thalamic activity can certainly steer around the cortical activity, and it's not useless. Actually, you can see that it's actually affecting the arm, where it causes this major sort of blip in the arm trajectory for the 4 hertz, and the 10 and the 40 cause very aberrant movements. All right, so thalamus can steer around the cortex and the arm, but does it actually do that in real behavior? Well, one prediction you might make is if it does that in real behavior, then there should be real rich information in motor thalamus about motor cortical's activity. So in order to look at that, what we wanted to do is record from both structures, from thalamus and cortex, and then relate their activity patterns to one another. Now, this ends up being one of the most difficult experiments we've ever tried, because you need to find the right parts of motor thalamus, and that's not easy. So in order to find those parts, what we leveraged was the use of optogenetics in these neurons, and these new electrodes called neuropixels, which just give us a lot of chances to find the right units. They have tons of electrodes on them. So basically what we do is we express channelopsin in thalamus with an electrode into thalamus. We shine blue light onto cortex, backfire these neurons, and hopefully we'll see them on the end of the electrodes. This works sometimes, where we can see every time we apply the blue light, this bank of electrodes here is getting antidromic spikes. So now what we can do is we can take those identified units and then relate their activity patterns to what we see in motor cortex. And so that's what I'm showing you here. This is the population activity trial average for thalamus and cortex And you can clearly see qualitatively, these things are very, very similar. Actually, it would be hard to know which one's which if I didn't put the labels on there. Now, that's a qualitative assessment of this, but we can actually take one step further. We can ask actually how related they are by asking if we can predict the cortical activity from the thalamic activity. Okay, so we're going to take a regression-based approach. We're saying, if I know what my thalamus activity is, can I predict the future state of cortex? And for comparison purposes, we're going to actually ask the same thing for cortex. If I know where cortex is, can I predict the future state of cortex? If it's an autonomous system, it should do really, really well in that situation. But what we find actually when we do this regression-based analysis is thalamus almost always does as well at predicting the future state of cortex as does cortex. In some cases, it even does better, as what I'm showing you here. And all this really comes down to is suggesting is that thalamus has rich information about the cortical activity. And taken all together, what we think that this really suggests is that unlike the role of the spinal cord and things like locomotion, motor cortex is not an autonomous pattern generator for skilled movements, as I think is the leading hypothesis in the field, but rather it seems to be more in this distributed, input-driven configuration, where it's constantly relying on its inputs in order to correctly make this pattern that's so necessary and sufficient potentially for the movements. And what that really means for us, the big take-home message for us, is that means in order to really understand the neural basis for skilled movements, we're going to need to fundamentally understand how all these inputs are collaborating with motor cortex in order to generate this pattern of activity. Now, I was going to tell you about a major effort in the lab, which is now trying to dissect out all the different roles of the inputs coming into motor cortex. This is what most of the lab is working on. But I'm going to actually change it up a little bit for today, given the audience. And I'm going to tell you that one of the other things that we're doing, in addition to trying to understand the inputs to motor cortex, is understand its output. How does this time-varying pattern actually lead to movements? And of course, ultimately, what's going to have to happen is these messages, either directly or indirectly, are going to have to get down to the spinal cord for a forelimb movement. And they're going to eventually need to get to motor neurons. Okay? And we would really like to know, what is the logic of that descending control over motor neurons? Many complicated things could be happening here, because what is especially true in the mouse and is also majorly true in the primate is the cortex doesn't hook up directly to motor neurons. I mean, they do in part in primates, but hardly at all in mouse. And what they mostly do is hook up to these complicated interneurons sitting in the middle of the spinal cord. And unfortunately, what that means is that there's a polysynaptic pathway between the cortical spinal tract and the motor neurons for the most part. And as a field, we're particularly bad about tracking and tracing in an interrogate fashion across polysynaptic pathways. And so how can we get to this answer? Well, we reason that there is one really good, but very, very hard way to get this, which is by getting a full connectome of a hemisegment of spinal cord. And if we could get that, then we could trace through all these synapses and get the full sort of input-output map for motor neurons. All right, so this seems like a Herculean effort because basically it's about 1 millimeter cubed of connectome, which would put you in the biggest connectome collected. And it's not easy to get there. There's a $100 million DARPA project that created one of those. All right, but I think in the last few years, there's been a couple of advances that have now pulled this into the realm of very, very difficult, but doable. Okay, and the advances are these. The first advance is basically an advance on sectioning. So that used to be done basically on a microtome and was done by a human. It was very laborious, error prone, and you could hardly get anyone to do it, especially for a volume this big. So a major advance that was made by Harold Hess's group at Janelia over the last few years was actually to mill the tissue, to actually use focused ion beams or gas clusters to mill the top of the block of tissue and then to image the top or block face. And that removes this whole sort of manual sectioning element. Okay, so that's one major advance. The other major advance is actually imaging the tissue, which takes a long, long time. If you talk about a millimeter cubed with electron microscopy. And traditionally, this has been done with a single beam, and you sort of move it all across your specimen. But in the last few years, new types of scanning electron microscopes have been produced that have many, many beams all running at the same time. And this greatly speeds up your image analysis acquisition time. Now, these things have been great, and really, I think are going to push the field forward. But there's one more significant hurdle, which is sample prep. Like, you need to have a piece of tissue that's a millimeter cubed that actually looks good all the way through the millimeter cubed. So you need to have good fixation, good staining, and that is not trivial. And in my opinion, is the major bottleneck going forward. So we worked for a couple of years on redoing the entire sort of EM sample prep for spinal cord and for other tissues. And I think we've come up with a really new a modified protocol that allows us excellent staining and fixation throughout an entire millimeter cube block. Here's just a representative example. So now we're in the position where we can start to think about collecting volumes of electron microscopy images of an entire spinal cord. And we have now done our first sort of test volumes here. So this is a very large chunk for electron microscopy. And as we're going, basically this movie is showing us, we're sort of flying through the Z sections of this, basically with isotropic resolution through large swaths of the spinal cord. And so we think in the next few years, with a huge effort still ahead of us, both financially and labor-wise, we can get a connectome of a millimeter cubed hemisegment of the spinal cord. And that, of course, will allow us to track and trace between the cortex and the motor neurons. But it won't end there. I mean, once you have the connectome, you can sort of do anything with it. You can track other major descending routes, or look at the intrinsic circuitry of the spinal cord, pattern generators sitting in the spinal cord, or as my wife first started, trying to figure out how the superficial dorsal horn, with its sort of amazing collection of cell types, does processing of somatosensory information, such as nociception. Okay, so we, of course, are really excited about getting a connectome of the spinal cord, but once this pipeline is set up, I just want to mention, there is this, of course, real opportunity to also image and compare with diseased spinal cords or spinal cords after injury, and to look at all the details, not just even neuronal circuitry, but what happens to glia or to other factors, and see what has changed during these conditions. And I think it is the kind of data that we really need. Okay, so I'll get off my soapbox about that. And then the last few minutes here, oh my gosh, I'm almost out of time. I'll go through this very quickly. I talk too much. I just want to mention a brief story that we published during the pandemic that relates back to our interest in understanding how these animals acquire these skilled movements. That's one of the main motivations for studying skilled movements. And one thing that we think about for acquiring skilled movements is that maybe there are outcome systems in the brain that are monitoring the outcomes of prior actions and using those outcome signals to either fortify or to change the upcoming motor programs and motor plans. And we really would like to know where those outcome signals were. I mean, they've been studied in decision-making tasks, but not for the motor system. And so we thought that we should look for these in the motor system. And we thought motor cortex was a really good place to look, because the dynamics have to come through motor cortex. And of course, also that we think of cortex as a great learning machine. So we wanted to look for these outcome signals sitting in motor cortex. Outcome signals should happen after the movement and have information about the prior trial. All right, I'm going to go through this very quickly because I recognize I'm out of time. The first thing that we're going to switch from electrophysiology to imaging. We're going to use genetically encoded calcium indicators to monitor these things. The first things we looked for were there were outcome signals in these layer 5 pyramidal tract neurons. We labeled them using this virus that we helped develop called AAV retro. Gets picked up by the axon terminals of neurons. So if you inject it in the brainstem, it will basically only get picked up by the pyramidal tract neurons. And we looked for these outcome signals in these neurons. And long story short, they don't really have any. So if you look at cells and their activity after success trials or after failure trials, there's not much difference between these after the movement. There's some differences during the movement, but not much after, okay? So that was a little disappointing, but there's lots of other neurons in motor cortex to look at. And one set of neurons that we were interested in were these layer 2-3 neurons, which are a major class of excitatory neurons in motor cortex, but we had very little recordings from them because silicon probes don't really see their signals very well. So we did the calcium imaging. This is their average activity across all trials. Pretty disappointing. If you look at, these are a couple hundred neurons, not much is going on. But long story short, when you look at them for outcome signals, if you separate your trials for successes and fails, you get these beautiful outcome signals in these neurons. So this is a success neuron, long calcium transients only after success trials, not after failure trials. This neuron's a failure neuron, only shows these long calcium signals after failure trials. And I'm going to skip this part, but we really were interested in what the nature of these signals are. They are not reward signals at all. They don't depend on the reward whatsoever. And I'm going to skip those. And I'm going to skip this, but basically just give you the punchline, which is that we know that these outcome signals are actually remembered by the system. They seem to be generated in layer 2-3 in a layer specific fashion. But by the time the next trial comes around, that outcome signal is actually sitting within these layer 5 pyramidal tract neurons. So we got this outcome signal born in layer 2-3, and by the time the next trial comes around, it's determining the initial state of the network in layer 5. And so just as the last sort of little bit of data I'll give, we were quite interested if that actually mattered for the animal, whether these outcome signals actually were important for learning and adaptation of these animals. So we developed this little assay where the animal would have to adapt on the rig. Basically, all we're going to do is move the food pellet from one position to another position. They've memorized it here. And once we move the food pellet, they'll have to move their hand over to reacquire the food pellet in this new position. You can see the success rate gradually ticks up here over the course of 40 trials. And so now, in order to test whether these outcome signals are actually important for this adaptation process, during the intertrial interval, when we think it's moving from layer 2-3 to 5, we're going to scramble cortical activity, but let behavior happen completely unperturbed. And when we do that, you can see that the animals are no longer able to learn. So we think that motor cortex has these access to these outcome signals. They're found in a layer-specific fashion that evolves temporally. We think that they're not related to reward, but more something about the performance of the task, and that they're critical for the animal's ability to learn and adapt. And I think this is potentially, at least a hypothesis that we have, is access to these outcome signals might be the reason why neural activity in this behavior has to even come up to motor cortex. You want access to these little learning rules given these outcome signals. And over time, but basically the idea behind the lab is to understand skilled motor control, understand how all the parts of the nervous system interact in order to pull this quite exquisite behavior off. And then just the most important slide, the people who did the work, Jay and Jeremy did most of the experimental work for the cortex story, where Britton and Matteo did a large part of the analysis. And Jackie Schiller and Marie Lavzin were in my lab as a sabbatical, and they did the work on the outcome story. Thank you so much.
Thank you very much, Adam. This is really great. It's just amazing. Any questions? Anybody has any questions? I wanted to start with one question. So there is these projections to the thalamus, and seems to be a totally separate kind of neuron in layer 5, right? Can you tell us a little bit more about it? Any, how so if thalamus is so important in communicating, is that those particular neurons have some kind of pre-information, perhaps from those layer 2 and 3, learning the rules?
Yeah, so what Pandela is asking about is there was a story from Carl Sabota's lab about a particular class of layer 5 neurons, which seemed to have a dedicated line back from motor cortex back to motor thalamus. That is just one of the classes that completes that loop. There are, layer 6, for example, goes back to thalamus, and there are other classes that have collaterals. It's just that was kind of a special class that seemed to be dedicated for that. And, admittingly, my lab understands very little about that projection back. And one thing that we are trying to understand is how turning off motor cortex in general affects motor thalamus. Just early days of what we're doing there. But we don't have any real information about the specific class of neurons that project there. However, what we speculate is that this loop between thalamocortical, which we call the little loop, there's a big loop. The biggest loop is, cortex in the world that involves thalamus, but then there's this little dedicated loop that Carl has talked about. And we think that could potentially have very interesting sort of roles for maintaining activity patterns or building up activity patterns. We just lack the tools that really allow us to crack this. So even in Carl's paper, when they tried to perturb that particular small class, the effects on behavior were basically non-existent. And it's probably because that's just one set of cortical thalamic projections. And the rest of those, because they project to a bunch of other places, it's very hard to take those out specifically without affecting the other regions. So we don't have good information yet about what those cortical thalamic feedback systems are, unfortunately.
Thanks.
This is Vance. I'd like to ask a couple of questions, but first I want to tell you that your talk was fabulous. I love your data visualizations. It makes it possible for senior citizens like me to start to understand what you're talking about. It was really wonderful. What part of thalamus were you recording in?
Okay, so this is the part that I skipped because I wanted to get on my soapbox about spinal cord connectomes. So we are recording from motor thalamus, which is okay. So that's a bad term. And so there's 3 major components of motor thalamus, which are, they've been called many things, but let's put them in some anatomical terms. Ventral anterior thalamus, which is thought to get a lot of inputs or biased inputs from basal ganglia. Ventral lateral thalamus, which is thought to get a biased input from cerebellum. And then there's these interlaminar midline structures, which also project. So VA and VL sit right next to each other. So we shove our electrode, hopefully on most experiments, through both of them, okay? But that does open up this question of, okay, well, which one's doing it? Like these are very different systems. And so this is what half the lab is working on right now. We really wanted to know the answer to that, but we lacked a toolbox to separate out VA and VL. They sit right next to each other and the mouse are interdigitated. We didn't have a genetic access to those. So my lab, in collaboration with Sasha Nelson's lab and Josh Dudman's lab, did a full transcriptional profiling of the entire mouse thalamus. We found genes which would label VA and VL. We made transgenic lines to gain control of those. And now we're directly testing those. And so in work that's ongoing in the lab, I can tell you now, we didn't know this before, but ventrolateral thalamus, this thalamic nucleus that gets a lot of input from cerebellum, has a huge role in setting the dynamics of motor cortex. If you screw up ventrolateral thalamus, cortex, it looks like the total perturbation of motor thalamus. So it is obviously absolutely critical for determining the dynamics in motor cortex. Ventral anterior thalamus, which has been given tons of very interesting roles in action selection and vigor and things like, just tons of really cool speculations about what it might contribute in this basal ganglia cortical loop, so far has done absolutely nothing, like literally no effects. But we think that might be because that it might be playing a role on a longer time scale than the fine temporal time scale of the movement itself. And we're exploring that right now.
Okay, my second question is, I really thought that it was interesting, your failure and success neurons in upper cortex. Do you know if those kinds of cells exist in other parts of cortex?
Yeah, so I'm sorry, I went over this so fast. I must have been tough and slow today. In the decision-making field, people have looked at this way before us for decisions. And they have found neurons like this in more frontal areas of cortex, but generally they have been more of the reward absence of reward signals, like which ours are not. So I didn't show you the data for this, but if you give them a plastic food pellet, the signals are still there, so they can't consume a plastic food pellet. And if you actually let them lick for the food pellet, so they still get the reward, but they didn't use their hand, they're gone. So ours are very different than what has been found before, but we do think that there are other kinds of these outcome signals in other areas of cortex.
Murray, do you want to come on and ask your question? I'm going to ask Murray's question. First, he says,
I got it.
Okay.
Yeah.
Hey, that was amazing, obviously. For the connectome project, it seems really hard to choose which spinal segment to focus on. So what are you choosing, and what was the thinking behind it?
Well, I'm hoping we don't have to choose. First of all, I'm hoping that a pipeline gets created, and support in each way is provided. So we do many spinal cords. I think spinal cord is just an ideal volume for a mammalian connectome, because you can fit like a functional unit within the volume that is possible right now, which is millimeter cube. However, the first volume, which is really what your question's about, was not hard for me, because I really wanted to know the cortical inputs to the regions that innervate the forelimb and the hand. And so we picked, lower cervical levels for that. So a bit selfish.
Okay, but I mean, even within lower cervical, there's going to be, inputs to different motor pools and so on. Yeah. So it gets kind of fine-grained in there.
It gets kind of fine-grained, and in some cases, we don't have much choice. Like, you have to take like whatever, when you fix the animal, then you look with a high-powered X-ray, what chunk is like perfect without any cracks, because sometimes you get cracks when you do the plastic embedding. So we've sort of opened from the range from C5 to C8, and we'll sort of pick which volume looks the best for imaging.
Okay.
Yeah, but if you have suggestions of what the ideal one would be, we'll certainly listen, because we're still doing these test volumes before we like execute the big one. But yeah, I mean, it's going to be a little bit relegated to opportunistic sections.
Sure. Great. Thanks a lot.
Any more questions? I guess not. So I don't understand that. I mean, that's just a general idea. This idea of thalamus being a kind of driver of efference copy, right? So somehow, everything really goes through there. Is that something, is that something of thalamus? That is what you're really thinking? This idea of efference copy that people have talked for years?
Yeah. So as you know, like my whole Clark's column paper was inspired by the notions of efference copy. So for anyone who doesn't know, efference copies are copies of the motor command. They're thought to be used not for movements, but for perhaps guiding sensory processing or potentially planning next actions. They're in the most classical version, they're the little collaterals that come off of your motor neurons, or in this case, your upper motor neurons. And to tell you the truth, the whole basis of all the projects I've done in my lab in the very beginning were trying to like selectively remove efference copies and then ask what is the functional role of that copy onto the system, like whether it be used for planning or for sensory processing. My view of this has changed a little bit now, and it's changed in the way that I think efference copies are real. They're there, but basically trying to understand the role of one is difficult. We should, in part, like now, we're just taking the system as a whole, and we're trying to understand how. So the feedback coming into it is colliding with the feed forward sensory information. It's changing from that, and so efference copies are, or whatever you want to call them, the feedback from cortical to thalamus is a critical thing. But it seems like the more interesting or the more important thing is just the sort of activity along the full loop, which will, the efference copy is part of it, but it's kind of impossible to untangle it from everything else. And so now the emphasis has switched from efference copies to us from the information that is held separately, or at least, particularly for the different loops, whether it be cortical basal ganglia, cortical striatal, cortical thalamic, cortical body, cortical cerebellar. And so it has become increasingly difficult to think about efference copies in that way. Now, I will say, when a specific inhibitor of axon terminals really works, then I think you can disambiguate between what the efference copy role is. We're just not there, so I've sort of shelved it.
Thanks.
Yeah.
Any more questions? I guess no. So thank you, Adam. This is really an inspiring and wonderful talk. Just thinking from the field of spinal cord injuries, these are the things we need to do. But anyway, thank you. And I guess you're gonna talk to a few more people.
Yes.
About, yeah. Thank you.
Thank
And
you all.
I will email you. Thank you.
Yeah. Thank you all for coming.